can also hear me, right? Okay.
Yes.
So, Dr. Lucas Sjulson is a psychiatrist that works at the Albert Einstein School of Medicine in New York. He's not only a physician, but he's also a brilliant neuroscientist. And his major questions is to understand how the brain processes decision making, not just in a normal setup, but also in a drug addiction context. And he uses several methodologies to understand what is happening in the brain, not just in systems neuroscience, science, but also a computational approach or even other molecular approaches. But I will let him take the lead now. So if you can, I don't know if you would like them to interrupt you during the talk or if you prefer the questions only at the end, but
Yeah,
we can
please
do.
interrupt me during the talk. So I see all of you are sitting around sort of a big table, but I guess you're also then logged in. If you don't
Yes.
mind turning on your cameras, and then we can please ask questions.
Okay.
Otherwise, this will be boring. All right. So and I don't want I want you to try to learn stuff. So so please ask questions.
Okay. Thank
Oops.
you so much for being here.
Yeah, of course. And so yeah, so let's get started. Let's see, is this working? All right, so I'm just going to talk a little bit about the history. So what I'm going to talk about to start with is thinking about how do we think about activity in populations of neurons, not just single neurons. Okay. Um, so one of the very first people, I think the first person to record from an individual neuron was Edgar Adrian. This is almost 100 years ago now. And what he did, he built some sort of setup that's pictures on the right here, where he was able to record from an individual nerve, an axon, and then play it back through a loudspeaker. He's one of the first people to do this. And so I read this story that he wrote about the first time that he got this to work. And so he was working in his lab and he had some sort of frog and he dissected its optic nerve. And so the frog's eye is out and then there's the optic nerve. And then he has essentially a type of patch electrode type of thing stuck in the optic nerve where he could isolate one individual axon. And then what he would do is then he would hear like the click, 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 click when the thing is firing. And so his plan was he was going to get it in there and then he was going to go grab this lantern and then shine the lantern in the frog's eye and see whether there was more spikes. And so he had a lot of difficulty getting it mechanically stable. So he would bump the table and it'd go, and then he'd get it just right, and then it would shift again. And so he was really struggling getting this to be mechanically stable. And every time it would lose stability, it would just be and fire like crazy, and then he would stabilize it again. So what he did is he would stabilize it, and then every time he got up and tried to go grab the lantern, then it would go like that, and he would come back, and then it'd be stable again. And then when he went to get the lantern, it'd be, and then he'd go back. And this happened several times before he finally figured out what was going on, which is that when he was going to get the lantern, he was walking in front of the eyeball, and he was listening to the eyeball watching him. Right. So this is the first person on Earth, which I think is a pretty amazing moment to be the first person on Earth to experience something like that. So, but those are the very first recordings we're talking about, almost 100 years ago. Um, yeah, and one of the things he figured out from that, right, individual cells fire action potentials and they encode information. For example, when he would move in front of the eye, he could listen to the eyeball, watching his movement and encoding it as action potentials. Another thing that happened now a little bit over 100 years ago was Hans Berger. So he was a psychiatrist who was interested in the study of telepathy and telekinesis and these sort of phenomena that we now know don't exist. He apparently had no training at all really in electricity or physics or Ohm's law or anything like that. And he decided he was going to try to record potentials from a person's scalp. And so what he did here was to build this machine. And then what you can see here, this is one of the first recordings he did, where this is where a person closes their eyes. And then the electrodes over the posterior part of the skull pick up the posterior alpha rhythm, right, in a human. And so apparently when he first published this, absolutely nobody believed it. And there was this whole gang of physicists who said, you don't even understand Maxwell's equations. You can't record like that. The brain doesn't make these potentials. None of this makes sense. It's all artifacts and blah, blah, blah. And something about 5 years later, a few groups simultaneously published that they had all reproduced his results and he was completely right. So old Hans got the last laugh, right? So don't rely on expertise necessarily. But one of the key things I think that was maybe underappreciated at the time, or we should think back on and the importance of it, right, is that if you can measure potentials at the scalp, right, all the way at the scalp, through the skull, through the skin and everything, that means that individual neurons, their activity is highly correlated and highly structured. And the fact that it's highly structured is that all the potentials add up, you can see here, to something that's actually recordable all the way through the skull. Okay, so we know now that back 100 years ago, people knew that information was encoded in action potentials, and they knew that the activity of cells was tightly coordinated. Okay, so let's take a step back and think about, so why did nervous systems evolve at all? Someone answer this question. Why did nervous systems evolve, please? Someone answer this question so I can move on.
You can use the microphone.
Microphone.
Oh, someone's putting in
Like a form to adapt ourselves to a new context.
Sorry, what did you say, to a new what?
Like a form of adaptation to a new context always, so we can keep alive.
Yeah, I mean, that's all correct. I think I'm thinking even simpler than that. The answer that I would give would be movement. I recommend a couple of good books. One of them, Eye of the Vortex by Rodolfo Linus. So what I mean by that is that there are simple movements that animals without nervous systems can make, right? Because they just have a group of cells that fire or something like that. But there's certain movements that require coordinated firing of sequences of muscles. And swimming movements are one of those things. And so actually some of the first animals that had nervous systems were jellyfish, and they have that these muscles contract in a certain pattern which allows it to swim. And they're actually not, they don't have any eyes or anything. They're not aiming at anything. They're just swimming. They only have motor outputs. There's this great example, this animal called the sea squirt that Linus talks about in his book, where it hatches from an egg, and it's kind of a primitive invertebrate that kind of is, sort of has some vertebrate-like characteristics, and it's kind of like a tadpole, and it has a brain and a sort of spinal cord, and it hatches, and the thing swims around, right? And it swims to the place where it wants to go, then it latches down and attaches to a rock, and then it grows up into its mature form. And throughout this process of metamorphosis into its mature form, it digests its own brain, and then the adult has no brain, right? And the adult is basically kind of like a sponge, right? So that the juvenile form has a brain and resorbs its own brain because it doesn't need a brain because it's not going anywhere, right? It's not moving. Right. Another great book I'd say about this is a fantastic book written for a lay audience, but has a lot of interesting points in it. But why did nervous systems originally evolve? And the point is that that movement, I think a strong case can be made that movement was the original reason, and then additional things got added on later. And the logic behind this, and I apologize, it's kind of a low resolution image, right? Is that any sort of coordinated movement requires pattern sequences of action potentials. And so this is an example of, so what I was talking about, that swimming motions involve contractions of muscles in a particular pattern in order to cause a swimming motion. And if you record from the spinal cord of a lamprey, there's a classic experiment where you can see that those patterns exist even in the isolated spinal cord. Right. So the idea here that pattern sequences of action potentials, right, are what is needed. Okay, and on some level, this is kind of obvious, right? That if you perform some sort of movements, right? Like if I take my arm and I go like this and go like that and go like that, that there's a particular sequence that those things have to fire in. If I say a word, right? That it's obvious that on some level, if action potentials make a muscle fire, that in the brain there are neurons that fire in specific sequences which cause specific, motor sequences to occur. And so this is all kind of obvious on some level. So the point that I'm getting at here was that if you think back to 1930, almost 100 years ago, what was known at that time? Okay, number one, Lord Adrian had figured out that action potentials in individual neurons encode information, right? Another thing that was known is that neuronal activity is highly coordinated across the population to the point where it can actually be recorded from the scalp surface in a human through, quite a bunch of thick tissue. And it was known that the nervous system must use sequences of action potentials across different cells in a population in order to generate complex movements, which we're capable of making, right? And so the conclusion of this, the conclusion that I draw from this, which was known almost 100 years ago, is that really how the brain operates, it's really about how activity is coordinated in populations of neurons, not individual neurons. right? Any one neuron in your brain doesn't do anything important. Someone raised their hand.
Yes.
Yeah.
I'm Roberto. I would just to come back from the point of before about the why neurons have evolved. Just because I'm working with these animals that are ctenophores, and they are like the sister group of all other animals, like more ancient than sponges. And they evolved this completely different nervous system. For example, the neuron, they don't have synapses, they are just connected without any division. And probably they also don't have action potential, but still they can navigate in the sea. They can have some complex behavior. Yes, I will just to say add this point if you don't know about this type of animal.
Okay, thanks. Yeah.
Okay.
Yeah, so there's a lot of interesting stuff in the world in biology, right? But anyway, the point that I'm trying to get at here, I think, is that the larger point I think I was trying to get at is that we know that complex movements require sequences of action potentials. And I mean, yes, sorry, that some animals that are certain sizes don't, have graded release of neurotransmitter and so forth, but the larger point here is that people knew, even almost 100 years ago, people knew, if you look at the data, that really populations of neurons are what is important, right? Not individual, at least in mammals, right? Individual neurons are not important, right? And people in, theoretical neuroscience people in the 20th century, they knew this, right? And they thought about it this way. And so you've probably heard of Donald Hebb and Hebbian assemblies. And so the idea here would be that you have some, sparse population of cells that fire together, and that encodes an apple, or it encodes, the concept of an apple, the concept of your grandmother, things like this, right? And the changes in the weights between cells could cause different assemblies of cells that would fire. And so the idea here, it's not that this cell is an apple cell, right? It's that this group of cells is an apple assembly, right? And that an individual cell could participate in both an apple assembly and a banana assembly, right? But the point is it's the constellation of cells that fire, not the individual cells that matter. And this, of course, ideas of synaptic plasticity all come from this. And then John Hopfield, who won the Nobel Prize fairly recently, he was one of the first people to show that you can essentially store memories in a network of neurons like this, and that if you store a memory, and then if you give a partial input, that it'll essentially reconstruct the original one, right? So and it's not about any one individual neuron; it's about the population of neurons and the interactions between them. So what I'm saying is that this was known a very long time ago. Okay. So, however, what happened in the field is a little bit different. Okay. So what ended up happening in the field is that the technology at the time could really, when people started recording from neurons, in vivo, mostly in mid 20th century, they could really only record from small numbers of neurons, maybe only one, maybe two or three. And so they discovered a number of important things. And one of the first things they discovered is that cells in lots of parts of the brain have things that we could call receptive fields or tuning properties. You record from V1, you can say that cell responds to stimulus right there, but not right there. Right. So it's known the cells have receptive fields and tuning curves, right? So which is properties that they essentially, going back to what I was talking about before, they encode information about the outside world. So the discovery of this, tuning to oriented edges, this was Hubel and Wiesel won the Nobel Prize for this, right? They understand that you can have tuning for non-trivial stimulus features, right, encoded by individual neurons. Okay, and so I think people like Hubel and Wiesel and all these 20th century neurophysiologists, we all stand on the shoulders of giants, and these are the giants whose shoulders we stand on, right? But I think that what I would say, though, that there's a couple things that they weren't quite right about, right? And what I would say is that the technological limitations of the time really constrained people's thinking about how the brain operates. Right? And so, like I said, they could record from small numbers of neurons. And so here's another classic example that another Nobel Prize has won for. And this is place cells, right, in hippocampus. So this is if you have a circular maze, circular arena, and an animal runs around in this arena, and you just plot where the cells fire. For example, there's a cell in the hippocampus that fires there. There's one that's fired there, and those are what are called place fields. Anyway, this is another type of tuning property, right? That individual cells have. Another thing, which John O'Keefe actually discovered both of these things. That's the only one won Nobel Prize so far. The second thing that he discovered was something called phase precession. And so what that is that a receptive, so the place field is sort of like a receptive field in space where the firing rate of this neuron A increases as an animal goes into this place field, reaches a peak, and then it goes back down, and then it stops firing again. So this is a classical kind of notion of like the firing rate encoding the animal's location. However, another thing that Hopfield discovered is that there's a theta oscillation in the hippocampus of rats, and that if when the rat first enters the place field, that the cell fires at a relatively late phase of theta. And then as it progresses further and further into the place field, it fires at increasingly earlier and earlier phases of theta. And this is the phase versus the position. And so these neurons, they not only fire in certain locations in space, but the temporal structure of their firing relative to this external oscillation, right, actually encodes information. And so back in the 20th century, people talked about this a lot. You might have heard, you don't hear this as much anymore. People talk a lot about rate coding versus temporal coding, right? And the idea here is, well, are neurons encoding the animal's location of the firing rate, or are they also encoding it, you know, in the specific, very fine structure of the timing of the spikes? And phase precession was taken as probably maybe one of the most undeniable examples of temporal coding in the nervous system, right, where the exact timing of a spike matters. Okay, and so traditional analysis strategies for this type of data is that you traditionally bin action potentials. So the idea is that you have some sort of train of spikes where you have a time bin of a certain length. There's 0 spikes here, 1 spike, 0, and then you take this train of spikes and you convert it into a string of numbers. And then you model it as a Poisson process. So essentially, it's like a random process of how many spikes you have in each time bin, right? And then you calculate the firing rate. And when I say firing, some people say firing frequency, and this is a subtle thing, but typically frequency usually implies regularity. If something you know has a frequency of 4 hertz, it means it goes exactly every 250 milliseconds. But rate just means that if something is a rate of 4 spikes per second, that just means if you have 10 seconds of data, you have about 40 spikes, right? So it's not exactly, so just to clarify that terminology, people do things like look at the inner spike interval distribution, look at this, and there's all sorts of things people do to look at this. Autocorrelogram, so it's a cell being correlated with itself. And what you can see here is you have this big gap in the middle, which is because of an exponential refractory period, right? And these are all the sorts of analysis strategies that people, the 20th century and in the 21st century as well, people use to look at individual action potentials. You can also, when you report from small numbers of cells, there's a lot of different analyses that people have done that look at pairs of cells. So if you have 2 neurons at the same time, then you can look at interactions between the 2 neurons. An example would be if you have a putative excitatory cell that synapses on another cell, then you can see that when the first cell fires, that makes the second cell more likely to fire with a very slight time delay. A similar thing with inhibitory interactions between cells. Okay, so we can see pairwise interactions between cells that way. And what I should say is that, and this is true for anything in neurophysiology really, is that most of the analysis strategies really depend on correlating single unit data with something else, right? And so what I mean by that is it could be, you know, a stimulus on a screen, it can be a movement that the animal makes, it could be the animal's spatial location in a maze, right? But the point is that sort of, if you record from lots of parts of the brain, it's hard to understand what you see because it can't be correlated in some obvious way with an external variable that can either be observed or imposed, right? I think places like B1, you know, primary visual cortex or hippocampus or motor cortex, those areas we've made a lot of progress because there's some sort of external event or parameter that you can measure or manipulate that has a strong correlation with the activity of the cells, right? And so typical types of analyses that people use are things like peri-event time histograms, receptive fields, place fields, tuning curves, and information theory is a sort of big fancy formalism that people can use to quantify how well action potentials in the brain essentially are correlated with something external. And this has been the basis of a lot of what we know about the neurophysiology of the brain. And I should add that this is why some parts of the brain are very well studied and some parts of the brain are hardly studied at all. And it's because there's certain areas that are more favorable or more amenable to this approach, where this approach works better in some brain regions than others. And people also record local field potentials, which is not the individual action potentials, but the just background voltage. And yeah, people have looked at things like phase locking, phase precession, so the relationship between local field potentials and the action potentials. And you can also do this across, or you can look at local field potentials in 2 different brain regions and how they synchronize and this sort of stuff. Okay, so but the point is that just recording individual action potentials doesn't really tell you anything. It's when those action potentials have some sort of relationship to something else is when you can figure out something about what's going on. Okay. I should add that the simplest models for this type of stuff are just linear models, essentially like linear regression, right? Where you can just model the firing rate of a cell as some coefficient times whatever your stimulus is, plus a running speed term, plus a baseline firing rate term, and then some sort of error, right? So really things like linear regression are kind of as simple as it is, are really the basis of understanding the relationship between the firing of the cells and some other thing, right? And so one of the problems with this type of thing is that the firing rates are not normally distributed. So this linear model implicitly assumes that firing rates, for example, can take negative values. And firing rates, of course, can't take negative values. So, which is the most obvious violation of the normality. And so the sort of extension that you see people using more and more, for example, of things like GLMs, generalized linear models, where essentially it's just a nonlinear function of the, it's the same thing as linear regression, except it as a nonlinear linked function here. And so GLMs, despite their name, generalized linear models, are actually a nonlinear type of model. And the one that you most commonly see is natural log. So that's appropriate for a Poisson process. So the point is being that you can now model in a slightly more rigorous and quantitative way the activity of a neuron as a function of these different things that you can measure or manipulate, right? And understand the relationship between those. So yeah, and then I should add that there are now these sort of even fancier methods, kernel regression methods, where you can actually do things like model the time varying responses to the stimulus or the running speed, right? But they're all kind of extensions of the same basic model where you're taking some sort of activity in the brain and you're correlating it in creating a model that correlates it in some way with something else that you can observe or manipulate. So before I move on, does anybody have any questions about this?
Yes. But more than a question, is just to see if I understood. So basically, you're presenting linear models that are for samples that are normally distribute, right?
Yeah.
Okay.
Linear models, like linear regression implies implicitly that things are normally distributed.
Okay, so and it doesn't fit with the real life of neurons because firing rates cannot be I mean, the distribution it's not normal, right?
Right. Yeah.
Okay, so you presented the generalized linear model that it's something that could be that could fit more with the nature of neurons. But then the kernel regression is because we can add also the time has another variable, right? Just for me to understand.
Yeah, exactly. Yeah. So if you look at the firing, what it's saying here is that the firing rate here is somehow related to the stimulus. The firing rate at time t is related to the stimulus at time t, but maybe the firing rate at time t is related to the stimulus at time t-1 or t-2, right? And so so that's the kind of thing. There's these more complicated regression methods that can, find more complicated relationships between these things.
Okay, thank you.
Yeah, but I think the bigger point that I'm trying to make is that there's a lot of effort has been expended in terms of figuring out ways that you can take one neuron in the brain and then somehow correlate it with some other thing that you can measure or manipulate. And then most of our understanding, of the brain is really oriented around that type of thing, right? If you think about it, this is a big picture concept, right? So the problem, as I talked about, what was known almost 100 years ago, right, is that neurons in the brain do not operate independently, right? And I should add that it's not that people didn't know this. People all knew this, but I think that they were kind of forced to operate as though it were true that they did. And so this is, putting aside, Hans Berger's work from the 1920s and so forth. Remember this paper coming out, 20 plus years ago, And it was pretty striking at the time, where what they did here was to they did intrinsic, this is a monkey, I believe, and they do intrinsic signal optical imaging, where they essentially optically measure blood flow, and then they record from one cell, and this is in visual cortex. And what they find is that if you essentially look at the average pattern of the intrinsic signal, every time that the cell would spike, right, that you would see that it's highly non-random, right? That so essentially that this cell is participating, right? It's like it's a member of an orchestra. And if you look at just every time that cell fires, this is what the other cells are doing. Okay, and this is what happens when it's a stimulus evoked spike. So interestingly, so what does it look like if the cell just fires randomly? So this could be the point is this all could all be just because the properties of the stimulus make it look like this, right? If you had a different stimulus, it would look like something different, right? But it turns out that if you look just spontaneously, when there's no visual stimulation and a cell just fires, whenever the cell randomly fires, that's what all the other cells are doing, right? So this shows what we, on some level, already knew, right? The cells in the brain are tightly coordinated. They're not operating independently, okay? So, and this sort of stuff was known, and people did think about it. So back in the 1980s, this guy Georgopoulos was recording an M1, and what he found is that if you have a monkey that moves a joystick in different directions is the task, and then if you look at which direction the monkey moves the joystick in, that the cells in motor cortex would fire a different amount, and they would have a certain sort of tuning curve similar to orientations in V1, something like that. And so what he did was he recorded many cells, not at the same time, but just like on different days, right? And if you just over many days, you just get a huge number of cells that you have recorded. And if you lump those all together and pretend they were all recorded at the same time, which I'm calling it a pseudo population, because they didn't have the technology to record a whole bunch of cells at the same time. But they could record them one at a time and then put them all together into a big population. And what they found is that they could essentially decode the animal's reach direction very accurately. Each individual neuron was not accurate, but the whole population was actually very accurate in terms of its encoding of the reach direction. And so the big idea here, right, is that information is encoded by a population of neurons, right? And so I'm saying this was known a very long time ago, but just the technology at the time made it very difficult for people to even though they knew it, that all the analysis strategies and all the papers and everything really are oriented around single neurons, because that was what they could do. Okay, so now we have new recording technologies, right? We have things like tetrode drives where you can, put dozens of tetrodes on the head of a mouse or a rat. We have silicon probes. So this is the NeuroPixels 2.0 probe. And we have things like calcium imaging, right? So all of these things, and there's pros and cons of all these things, right? Tetrodes, I think some of the best things about tetrodes is if you want to record from multiple brain regions simultaneously, you can configure a tetrode drive to sort of put them where you want. Silicon probes, they have a higher site density than tetrodes, and they have a fixed geometry, which can be very valuable if you want to record and sort of know, that if you have 4 shanks, that this one is exactly 200 microns to the right of that one and so forth. So you have a better knowledge of where the different sites are than with tetrodes. Calcium imaging, of course, you can see the cells, might be helpful to identify cell types. It's easier to track the same cells longitudinally, but it has lower temporal resolution. But I think that another thing that is maybe under-recognized is the calcium imaging. So things like silicon probes and tetrodes, you can just record for 12 hours. And with calcium imaging, you're limited to much shorter sessions because of photobleaching. Anyway, but the point is that all these things have pros and cons, but we have multiple different technologies now that could allow us to record many neurons at the same time, okay? And as a result of this, because of the technology has changed, our conceptual thinking about how the brain operates has changed, which is somewhat unfortunate, but it's just a reality of humanity that that's how we work, right? So there's what I would call sort of the old 20th century way of thinking about brain activity, and then the way we should think about it in the 21st century, right? And I think the way people thought about it, right, single neurons encode information, and populations are made out of a bunch of single neurons, right? What I would argue actually is that single neurons don't really encode information. It's that neuronal populations encode information. And if you record one neuron, you're just sampling one of those neurons. But it's the population that comes first. The population is what does it. And we can just sample one cell from that population. Okay, and at that time, I think they just wanted to record from single neurons to see what do they do. This neuron has, place fields. This neuron is tuned to this orientation of a visual stimulus. They wanted to understand what do individual neurons in the brain do. Okay, and I think where the field is now, where the field is going, is that we can't record necessarily from all the neurons, but we want to be able to record from enough of the neurons that we can make an inference about what the entire population of neurons is doing. Right, so I'm saying that this is even though the technique is ostensibly the same, that the actual questions are different now, right? And the conceptual idea of what we're looking at is different. Okay, I think an old way of thinking about it, people are like, oh, you can record more if you record 100 cells at the same time, then that's the same. Then in one day you could record what used to take 100 days, right? And now I think that what we're thinking more is that recording a bunch of cells at the same time is actually qualitatively different. It enables you to understand things that you cannot understand by recording one cell at a time. Okay, and I think one thing I would go so far as to say that if we just recorded one cell at a time in the brain of animals or humans or whatever, we could do that for 1,000,000 years and we would never understand how the brain works, because the coordination between the different cells is such an important ingredient of what the brain does. And something you see people have said in the past a lot, which I completely disagree with, is the notion that neuroscience is so-called data-rich but theory-poor. We don't have a lot of theories in neuroscience, but we have all this data and nobody can make sense of it. And I actually think it's the opposite. I would say that neuroscience, compared to a lot of other fields like physics and chemistry, neuroscience doesn't really have a lot of good theories because historically it hasn't had high quality data. There's been a lot of low quality data, lots of recordings of one cell at a time, but it's only relatively recently we're starting to get really high quality data. That's the sort of thing that can drive development of actual theories about brain operation. And I'm going to talk a little bit more about that in the future. Actually, one thing I want to say, which is that there was a conference, it was about the year 1999 or 2000, where people talked about, there's this new technology where people are going to be able to record 100 neurons at the same time. And this is something my lab does completely, we record more than that every day, right? But they're like, someday in the future, people record 100 neurons at the same time. And so they asked people at the conference, what would you do if you had that data? And a guy got up, went to the microphone, and he said, well, I would throw away 99 of those neurons and analyze just one, because I know how to do that, right? And it was a joke, but it was kind of not a joke. I'm saying this is what I'm getting at, is that it requires a different type of thinking and different tools and technologies and frameworks to analyze simultaneously recorded cells. Okay, so here's another point. So remember I talked about theta phase precession? If you record one cell at a time, what you see is that you have this oscillation, and that the phase of the cell's firing gets earlier and earlier relative to that oscillation, okay? Now, that's what happens when you look at one cell. You see evidence of temporal coding, meaning that the precise timing of that cell encodes relative to this LFP oscillation encodes information. This was taken as a big idea in the field at the time, temporal coding, okay? So what happens if you have more than one cell? If you have cell A gets earlier and earlier, cell B gets earlier and earlier, cell C gets earlier, cell D, what you're seeing is that on each sequence, you get ABC, ABC, BCD, CD. So what's happening is you're actually seeing that these place cells go in this order of A, B, C, D in space, and that sequence is being played back on each theta cycle, okay? And so these are what are called theta sequences. When people started to be able to record many cells, they could see these things. And so this maybe not everybody agrees with what I'm about to say 100%, but I would argue it's true, that what the original phenomenon of phase precession is really an artifact of single neuron sampling of theta sequences. That because we could only record one neuron, we thought it was this, but actually it was this, right? And it's something that's qualitatively different. The notion that cells fire sequences that essentially predict where the animal is going. And that's what we now believe the hippocampus does. It's not just encoding your location, it's encoding trajectories through space of where you're thinking about going, right? And recording one cell at a time missed that. And now we sort of know this is how it works. So this is a different perspective. Here's another interesting paper that came out recently where they trained animals to distinguish these two different gradings. And what they found is that if you look at individual neurons in V1, they have orientation tuning, and that the orientation tuning changes as animals get better and better at learning to discriminate specific orientations and not others. And what they found essentially is that if you look at individual neurons, they cannot make sense of the patterns. But if you look at how the populations are encoded, then what they find is that the changes that are made here are essentially orthogonalization of the vectors at a population level, right? So this is something that cannot be understood at the single cell level, yet is very clear. Is very simple to understand at the population level. Okay. So this comes back to my argument. The populations are really what we need to be studying, not single cells. Okay. There's lots of other ideas. One of them is called mixed selectivity. And this is the idea that individual neurons encode more than one thing at the same time. That might encode the animal's movement, the perceived value of the stimulus and these things, but they encode nonlinear combinations of those things. Right. Another thing is that population representations are actually different than what you get from individual cells. So a classic example of this is true in different sensory cortices. So if you measure the receptive field of a cell in V1, and this is also true in auditory cortex, and I think some other areas, and if you give a complex naturalistic scene and you record many cells and you look at their tuning, that the tuning of the cells is different than what you would expect it would be based on the individual receptive fields. Okay, because cells interact with each other. And so the idea here is that its population representation is actually different than what you get if you just essentially record one cell at a time. Okay, so how do we deal with this? How can we solve the problem that this guy 20 plus years ago, how do we not throw away 99 of the neurons? And so all of the approaches that I'm going to talk about, fundamentally all are grounded in linear algebra, okay? And some of you have probably taken linear algebra, some of you probably haven't. So my personal recommendations is that in science in the year 2025, you all have, this is all stuff you have to learn. It's not that hard, honestly, but it's important to learn. And I can give you these recommendations later. I think this is a very good book. And there's an online course on MIT's website where you can watch the whole course online. I think this course is very good. And there's also one on YouTube where they have animations that sort of show how it works. And so this is what I recommend for those of you who have not studied this. This is what I'd recommend a good starting point. Okay, so learn it. All right. And I should add, this is the basis of all, like if you think about anything where you have a lot of high dimensional data, like single cell RNA sequencing, things like that, they're all based on some level, they're all grounded in linear algebra. Okay, and so the idea here is that instead of just looking at one cell at a time and looking at its firing go up and down, the concept is that we can think about population activity as a trajectory in a high dimensional state space. Yeah, so this video is broken. Sorry about that. It usually works when I play it the third time. Yeah, so if you have 5 neurons here, and here's just visualizing 3 of them, right? You can see that the activity of the cells is of individual cells reflected here in this space, and you can see as an example there. This one goes up and down 5 times, and that's what's being shown right there. Right. So we can't imagine necessarily if we record 100 neurons this 100 dimensional state space, but that's a good way to think about how we can visualize and think about the population, the activity of populations of cells. Okay, so something interesting that oh yeah, so what's the point of this? Why would we do that first, I guess? So another thing is understanding computation's dynamics in a state space, by which I mean that instead of just understanding what single neurons do going up and down, understanding the space as some sort of structure that sort of pushes the trajectory around, right? And characterizing that, and there's whole field of nonlinear dynamics, this whole field that sort of operates on that type of thinking, right? That gives us a tool we can use to conceptualize and understand how circuits in the brain operate. Another thing, the next thing is dimensionality direction to find so-called manifolds. I'm sure most of you, 2-3 years ago when I talked about manifolds, nobody knew what I was talking about. Now I think most people know what that phrase means. The idea here is that if you had 3 cells here, in principle, you could imagine that trajectory would go absolutely anywhere it wants. But in practice, what you actually find is that the trajectories of these things actually are restricted to lower dimensional spaces in the full space. And these things are what we typically call manifolds. So in this case, we have a 3-dimensional state space where we have 3 neurons, but you can see that the activity really is restricted to this 2-dimensional manifold plane in this case. And that's because the different cells, their activity is strongly correlated. Okay. So what does low dimensional mean? So what it is low dimensional essentially means dimensionality is, you can think about it as the number of numbers that you need to describe something, right? So in this case, and the simplest method we'll talk about is PCA, principal components analysis. The idea here is that it allows you to rotate the axes to find axes that account for more of the variance. So in this case, what we could say is that this data is low dimensional, and we have the two cells fire so strongly together that we really have, the data is restricted mostly to this one-dimensional manifold here, right? High-dimensional data, where there's no relationship between these things, we can't really find any pattern like that. And so we can look here at the first and second dimensions, how much variance they account for. And this is the way of quantifying the dimensionality of something. Yeah, so here in this case, we can find some rotation that's slightly better than chance. What we can see here is that the amount of variance explained and how quickly it drops off as a function of the number of dimensions is a good way is one of the ways to quantify dimensionality. Okay. And this is an example of what real data looks like, where you have over 100 dimensions, and you can still see that there is this, now that there's a lot of the pink ones that have more variance than chance, and you have ones that even have less variance than chance. But the same sort of idea applies to when you have many, many more dimensions as well. So before I move on, what questions do people have about this? I'm going to talk a little more about PCA, by the way, how it's calculated.
Yeah, I think nobody has questions now.
Okay. Yeah, yeah. So the first time that I saw this sort of thing, I should add, was and actually the first field that really started thinking in these terms was really the olfaction field, and people doing olfaction in Drosophila and zebrafish and locusts. And this is an example where they record from many different neurons. And this is in zebrafish olfactory bulb, and they would give all the different amino acids in solution. And what they would find was that the trajectories or the odor representations for chemically related amino acids would cluster together in this PCA space. Right. So over 20 years ago, this is how people in the olfaction field were thinking about tuning of things, not in terms of the tuning properties of one cell, but in terms of in a way, the odor identity manifolds, right? And so how does PCA actually work? All right, so the first thing you do, I'll talk, and this is not something I can just explain. It's something you're going to have to go watch YouTube videos and really understand if you haven't learned it. But the first thing you do is you take your data and you put it in a matrix in the linear algebra sense. You say you have n neurons and t time bins, right? And PCA is basically a matrix factorization, by which I mean that it takes a matrix and it splits it up into several matrices multiplied by each other. Okay. And unfortunately, I don't have a great illustration in this. This is a whole topic. There could be a whole lecture on this. But a way of thinking about this, this one wasn't supposed to appear, is that if this is your data, what you can see is that this data up here actually, there's 4 different cells here, but this is actually made up of 3 different patterns. Okay, and these neurons here mixes the different patterns. So if the first pattern is this one here, where you have the bump there and bump there, you can see that the first neuron has basically 2 of those. The second neuron here has 0 of that pattern. The third one has 1, where you can see it's sort of, there's a bump there and a bump there, right? And this one also has it. So neurons 1, 3, and 4 have non-zero amounts of that pattern, right? Now the second pattern is this bump right there. And you can see that the second neuron here has a negative amount of that, and then the fourth one has a positive amount of that. But anyway, that's the idea, is that you can find that there's a smaller number of patterns that actually explain your data. Okay, and this is entirely data driven, where you take the data and feed it in, and then it finds the best representation. And the way of thinking about this is that as something called an autoencoder, right? Which is basically, if you have a neural network where you have your inputs here, imagine you just had 4 of these, and then there's a bottleneck where you put it down to a lower number and then expand it back out again. Okay, now if all these units were linear, and they're basically just, each of these things is just linearly summing its inputs here, then this basically would be PCA. That you're figuring out a way to squeeze something through a bottleneck of a smaller number of eigenvalues, and essentially it allows you to extract what is the smallest number of dimensions you need to reproduce the original pattern as close to approximate the original pattern as well as you can. Okay. But so the point is that this is all linear, and this is what's called dimensionality reduction, because in this case, you're taking a bunch of dimensions and you're reducing the number of dimensions. Okay, and one of the reasons dimensionality reduction works to some extent in the brain is because brain activity is lower dimensional than expected by chance. And that's an empirical fact about the brain. Okay, so PCA, it's a linear technique. One of the limitations are spikes are binned. That's a whole thing we don't have to get into. Again, there's this assumption that the number of spikes per bin is normally distributed. The cells can have negative firing rates. So it treats increases and decreases of neuronal activity as being the same thing, when in reality, those two things are not really equivalent. Positive and negative correlations are treated equally. So there's lots of limitations. The biggest one probably is that it's linear, right? So if the manifold is somehow curved, it can only approximate that as sort of two flat ones, like that, but it can't find curves in the manifold. Okay, it also doesn't handle sequences. So there's lots of limitations, but also lots of pros, advantages. One thing is it only looks at geometry, not the dynamics. So what I mean by that is that if you were to just shuffle the time points in your data, you would get exactly the same answer with the time points shuffled. It doesn't actually look at what happens at time point t, t+1, t+2. And there are versions of things like PCA that do, that actually look at things like dynamic mode decomposition. So there's a whole family of these dimensionality reduction techniques. So it's a huge topic that you can read up on. Another thing is nonlinear dimensionality reduction. And so this is an example of something PCA would do a very bad job of understanding that this is all different versions of the same letter, just warped and twisted and rotated and scaled. But nonlinear techniques can actually find the manifold there, which is curved. So there's a whole field, right? People coming up with dimensionality reduction techniques. One of the ones that you should be aware of is something called autoencoders. So remember I said that PCA is sort of like this, where you have these linear units, and then you pass it through bottleneck and you expand it back out. And so if you wanted to take your data and reduce it to 2 dimensions, that's how you do this, right? But so you can also do this using nonlinear neural networks. And so that's what people typically call autoencoders. And so this is a strategy to reduce to, for example, 2 dimensions that by using nonlinear transformations, right? So that's one that you should know about. Another one you should know about is techniques called t-SNE and UMAP. And these are essentially nonlinear dimensionality reduction techniques that reduce things down typically to 2 dimensions just for purposes of visualization. And these techniques have come under. Yeah.
Sorry for interrupt. I have a question, but how you can be sure that although you are reducing the dimensionality, you are preserve the dimensionality? I mean, can happen that reducing the dimensionality, you can miss something when you're going to decoder then?
Yep, that's absolutely right. Yeah, so in the case of autoencoders, the way it typically works would be that you train, that you start off with your input data, and then you essentially train this network so that it finds the weights where the reconstructed data matches the input data, as closely as possible, usually in sort of like sum of squared error type sense, right? Where just you try to get the closest approximation, right? And I guess what I'm saying is that even if the data actually had 4 important dimensions, you could still squeeze it down to 2, and you would still get some sort of reconstruction that was still better than chance, right? So this doesn't necessarily find the true, underlying dimensionality of the data. And yeah, there's always some distortion and so forth. And what I was about to say is that these techniques, UMAP and t-SNE, have come under fire a lot lately because you can't always squeeze things down. You can always squeeze things down to 2 dimensions and make a pretty picture and look at it. But that doesn't mean if the data actually is more than 2 dimensions, right, then you might be throwing out important stuff when you do this squeezing down, and it might be giving you an answer that's very misleading, right? So I think that these are techniques you can use to visualize things, and they're often helpful, but you can't rely on these too much, right? Because it's not everything just fits into 2 dimensions. And I should add, by the way, that I don't actually believe the brain activity is low dimensional. I mean, so that's a whole separate thing. So it is the case that brain activity is low dimensional in most parts of the brain, where it's mostly low dimensional, but there's some high dimensional stuff too, right? And so I want to clarify that I don't actually believe that dimensionality reduction captures everything that the brain is doing. But it captures a part of what the brain is doing, and it's important for us to understand it and understand what the relationship is between these latent spaces of relatively low dimension and, you know, things that deviate from that, right? So yeah, I mean, I don't have answers for you, unfortunately, about all this. These are
No, but
complicated.
it's okay. It's okay. It's okay. It also helped me to understand better. I mean, it's okay.
And I should say that this is not the sort of thing where these are like these answers have been worked out and everybody agrees on this. I'm saying this is kind of where things are at right now, right? That you can do these things, and is it the right thing to do? In some cases, maybe yes, some cases maybe no, but you should be aware of it. Is I think what I'm getting at. So this is another paper from 2003 where they used a nonlinear dimensionality reduction technique. And you see figures like this all the time in papers now, but in 2003 you didn't. And this is a good example of something where they would map out essentially an odor concentration manifold where you can give the same odor at different concentrations, and then you get these sort of curves that all fall in this low dimensional manifold, and the manifolds are different for different odors, right? And so this is the kind of stuff 20 plus years ago people were thinking. Now you see this stuff a lot, right? And it's a way of conceptualizing what the responses are, or you know, movement evoked activity or whatever it is, right? Here's another interesting thing that is a study from over 10 years ago now. But what they did was they recorded a bunch of cells in the brain of a monkey, and then they trained the monkey to I think originally started moving a joystick out so it could move a joystick to the different spots to get a reward or whatever. But then what they did was then they trained the monkey to, over time, stop using the joystick and just use and essentially just control its brain activity so it could move the things to the different points and then collect rewards. What they did was they mapped out there's an intrinsic, so here that they would report, say, 85 to 91 cells, and they found that there was approximately a 10-dimensional manifold that the activity tended to stay in. Okay? And so then what they did was they trained the monkey to move the neuronal activity to different spots on that manifold to get rewards versus trying to train it off the manifold to get rewards. And what they found is that it's very easy for the monkey to learn to control its brain activity within that manifold. But if they try to get it to make activity off the manifold, it was very, very difficult. And it turns out, well, what's interesting is the monkey was able to do it. It just took, instead of, you know, 30 minutes of training, it would take like 3 or 4 days of training. But the monkey could eventually do it. So what this means, who knows? Right, but the point is that this is something that's real in the brain. It's not just a description of brain act. It's not just a statistical description of brain activity. It's actually something that reflects something about what's actually going on in the brain. Right, the fact that there is a big difference between on manifold and off manifold movement. Right. So this is another thing that's you see a lot is that people talk a lot about things like representational drift, the things like place cells, or you know, tuning the individual cells tuning properties are not stable over multiple days, which raises lots of questions. But if you decode the population from a large number of sampled cells, then what you see is really that when an animal performs the same task. So this was recordings from a monkey that were made on day 1, and then I think they made them. It was like literally it was over a year later from different cells in the same brain region a year later doing the same task. And what they found is that that's how well the activity trajectories on the manifold line up, right? A year later, right? And so the population level representations of the manifolds can be stable even if each individual cell is not very stable. Okay. So once again, it's getting back to the idea of, and on some level of like, what other results could they possibly have gotten, right? If the animal knows how to perform a task and it performs that same task, there has to be something in the brain that's doing the same thing that was doing a year ago, right? And this is what it is. And neurons, manifolds can have topology intrinsically. So this is just an example from the head direction system where animals know which direction. Anyway, I don't want to belabor this one, but they can also have sort of interesting structure. Okay. So let's see. So one thing I want to say is that there's also, so what I was talking about, PCA is unsupervised dimensionality reduction. Okay, where you basically just take some sort of space and you just run an algorithm, basically splits out and says this dimension has the most variance. Okay, and then this is your first principle component. Or all those nonlinear methods, they just kind of just take your data and they split it up based on just looking at the data itself. Right. So there are other classes of methods which we call supervised dimensionality reduction, and these are things that where you also feed into the algorithm something in addition to just the data. And so what I mean by that would be, so say for example that that this would be like that an animal can get either like a red stimulus or a blue stimulus on the screen or something like that, right? And if the red activity patterns are what happens when it sees the red one, and the blue is when it sees the blue one. So PCA just breaks it up based on just what the data looks like. But there are these other techniques that essentially you tell it which ones are red and which ones are blue, and then it will find the dimensions that optimally separate the red responses from the blue responses, for example. Right. And so this is the whole thing where there's ways that you can do this, and you can look at, which dimensions in this space encode the identity of the stimulus, or encode the animal's movement, or encode the animal's belief about the value of the reward, and these sorts of things. And so the point is, this comes back to, remember I was saying that a lot of what we do in neuroscience is really about correlating neuronal activity to some sort of external variable that can be measured. Saying this is a way to think about doing this in high dimensional spaces. Right, that we don't have to just rely on these techniques. We can also figure out ways to find subspaces that actually have different functions. All right, what I've said, and yeah, and so this comes down to this whole sort of idea, right, where you're squeezing. So this model, an autoencoder, the question was asked earlier, so what is it basically doing? And basically, it's just training to find this reconstructed data that optimally that just matches the input data best. But you can also train it to do other things where, to make it so that, these units here optimally distinguish two different stimulus conditions or whatever it is. And often the answers are very different when you actually take that external information into account. Okay.
Luke, can I make a question?
Yeah.
One example, because PCA only reduce your data. One example, to split population is clusterize this with k-means or other things, okay?
Yep.
But PLSDA, you can do the cluster together or only to reduce the data?
Yeah, so the difference between something like k-means, right, is that k-means just looks at the data and tries to figure out how many clusters there are, right? And what PLS does basically says which of these responses were, had stimulus A on the screen, and which of these responses had stimulus B on the screen, and then it tries to figure out how to match that external variable to the data. Does that make sense?
Yes, because is it supervisionated? You put one label.
Is it what? I'm sorry.
You did form some label to algorithm learning or no? It's unsupervisionated.
It's supervised.
Yes.
Yeah, it's supervised. Yeah. So right, that's I guess what I'm getting at is that a lot of these methods people use are unsupervised, but people are more using supervised methods, and this, in a way, is what I'm getting at. That it gets back to this fundamental idea of somehow correlating some external variable with the neuronal activity, right? And these are kind of techniques that you can use to do that for, before I was saying, linear regression works when you have one cell, right? And these types of techniques work when you have 100 cells.
Okay, thank you.
Yeah, I mean, partial least squares really is just linear regression when you have 100 cells instead of one, right? So, but yeah, anyway, that's the point that I'm getting at, is that if you think about what we're doing, we're taking some sort of thing that basically kind of looks like a neural network, and we're in some way fitting it to the data, either just the data itself or some sort of external variables in addition to that. And then I'm saying mathematically that what we're doing kind of can be reduced down to something that looks like this. Okay. And so I guess PCA, for example, would take this and squeeze it down to 2 dimensions where the activity here matches the activity here as well as possible, right? For something like PLSDA would be something like it does it in such a way so the activity in this red cell is like strongest for when stimulus A is on the screen, and then there's the activity in this cell will be strongest, or this unit would be strongest when stimulus B is on the screen, right? But I'm trying to get an idea here that we're essentially training a model of some sort to have different weights to find the optimal representations we want. Okay. And so what I was going to say, the next step, which is what people are starting to do, and this is more sort of state-of-the-art, cutting-edge type stuff, is fitting models to the data that actually look more like neuronal models. So the stuff I was showing you before, originally PCA is just linear, and you can make nonlinear versions, but it's still an entirely feed-forward architecture, right? But you can actually make, these models where the cells, the units here interact with each other, right? Means you can take a whole bunch of data and then you can train your model to try to reproduce that original data. And the idea here is that you can actually train, models that kind of look like models of neurons, right? I'm saying this is kind of the next step to think about. The more complicated models that actually kind of look like neurons. Can we train these models to reproduce neural data and then analyze what's going on inside of the model? And then use that as a way of understanding this activity. So this activity here, you just have tons of this. It's very hard to make sense of it. But if you can figure out how to make a model that explains it, then maybe that model, is valuable. And this is an example of this where they can find this like low dimensional stuff in latent spaces and blah, 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 when they train models like this. And I should add that the current situation with this type of work is that I think people find it very promising, and there's a bunch of different groups doing this type of stuff. And one of the biggest problems I think they run into is that these models have lots of parameters. And so there could be several different models that look very different that all reproduce the same data. And so you could have 100 different models that all make an output that looks the same. And so the question is, how do you know which of those is the correct one? And so this is a sort of an ongoing area where this is not a fully solved problem yet. But I think this is the kind of approach you're going to start seeing more and more of in the future, right? Essentially using AI type techniques to understand our data by fitting models to it. And so I just want to close with this last statement, which I've heard people say this so many times, and I think it's just the wrongest thing that has ever been said. Okay, so look at ChatGPT, right? So ChatGPT has 175 billion parameters. So how much data did it take to train ChatGPT to make the model work?
Yeah.
It was 45 terabytes of data. Okay, and that's just ChatGPT, right? Now, just the brain of a mouse, I don't know how many parameters it has. Nobody really knows, but like it has more than 175 billion, right? And so how big does our training set need to be to make models of the mouse brain? And there's a lot of people who are like, oh yeah, you recorded 10 gigabytes of data. That's a huge data set. Neuroscience is data rich because we have 10 gigabytes of data. And I'm saying people are thinking on completely the wrong scale, right? And we need to be having petabytes of data, to be able to actually train models that can to adequately constrain the models so we can actually understand what the brain is doing. So I think that we're now entering this new age of not just big data, but of unimaginably huge data. And I actually think that amount of data will be required to be able to train these models and get interpretable answers. And so that's my opinion. All right. So just summarizing, populations are what matter, not single neurons, right? Things like dimensionality reduction and things where you can fit models to the data, which dimensionality reduction is doing that. I'm saying different ways to fit models to the data, I think, are really the path forward. Please learn linear algebra, right? Everything depends on that in terms of type analysis. And we're going to need a lot more data than we have. We already have a lot, but we're going to need a lot more. And that's my opinion, my belief. So anyway, yeah, I'm all done. Any questions anybody has?
Yeah, I do. Can you hear me?
Yes.
Oh, thank you for the presentation. It was very insightful. I do have two questions. They both have to do with the models themselves. One more with the models and the output it gives us, and one more related to data. So in terms of dimensionality reduction and all of these techniques and neural manifolding techniques, so they have been used a lot and they are still being used a lot. And you talked about how they are being developed into being more complex and into trying to capture and extracting more structures and dynamics. The thing is, there's a very famous, I would say, researcher called Mark Churton. He has been using these neural manifolding techniques, for example, for studying movement in macaques. And he came along with developing methods like JPCA, and eventually it was a big breakthrough because he discovered rotational dynamics in neural population recording and all of that. And then later in came Miguel Nicolelli and Lebedev, and they said, oh, well, we applied those methods to a lot of different data, and we always encountered rotational dynamics. So is rotational dynamics really a characteristic, or is it just coming up because of the model itself? So this would be my first question. So at what point are we actually developing models that are giving us outputs just because that's their endpoint. And the second question, I don't know if you want to answer right now, and then I can give a follow-up.
Yeah, let me just comment on that, right? So I mean, so the first thing about rotational dynamics, I mean, I think that's a controversial area, and I don't claim to know the answer to that. But it's part of what it's reflecting is the sequential nature of the neuronal activity, right? The fact that you have sort of this, so you can think about, these cells are firing first, and these cells are not firing, and then now these cells are firing, and that, so rotational dynamics are to some extent a reflection of the sequential nature of the activity, right? So I don't know what to make of, the rotational stuff, but the idea is that it's the rotational dynamics are capturing something that's a relevant feature of the activity that we know is a relevant feature. The second thing I would say about stuff like that can be a little bit of a caveat, right? Is that the kind of tasks that people like Mark Churchland are doing are typically low dimensional tasks. So if you have an animal just push a joystick in 4 directions or whatever, then the brain activity is going to be 4-dimensional, right? But that's not actually naturalistic complex movement that animals actually make. So I think that we need to be just careful about extrapolating from very low dimensional tasks or low dimensional stimulus sets to what the actual brain activity is like. So that's my answer.
Thank you. My second question would have to be also about the models, and when you talked about these models that are being developed, mostly about deep learning models and all these models that are being developed to analyze data. And you talked about how much data do we actually need to make these models work and for them to give us insightful outputs. So my question would be, are these models actually the way to go as for their capacity to extract patterns or structures in data, or aren't they going to be limited in terms of we don't actually know how these models fully operate in their processing of data, right? So are we giving them an input of data and then expecting an output, and are we considering that that's all we need to know in terms of, don't we actually want to understand what's in the middle, what's in between?
Yes, yes, absolutely. I think what I would say is that if you think about like the way that sort of like an early career, like someone who's very early in their scientific career, someone who's still a student, still learning, I think the way that they think about data, in many cases, is sort of like you look at the data and it gives you the answer, right? As this sort of thing where, I want to see if this, cells have fire in this location in space, and so you measure and say, oh yeah, the cells have a place field looks there, and the data gives you the answer. And that's kind of like, how science worked in the 17th century, whatever. But I'm saying that I think the way that on sort of a deeper level, the way that the relationship of data to truth is actually more the data provides constraints on a model, right? And then you have different competing models, and then you find data that constrains the models to help you determine which of your models is correct, right? I think that's how science actually works if you think about it more deeply. And what I'm saying is that we need to be more explicit in terms of our model building and our data for constraints. And so that's how all science works. That's how, Newton's, F equals ma works, right? I'm saying that the problem is that our models have to become more and more complicated, and our data sets have to become larger and larger in order for us to be able to say we have these competing models and the data constrains it and tells us which one is correct. The specifics of what you're, and I'm making just a very general statement, about that. And so in that sense, I do think that is the way forward, to make larger, more complex models that are constrained by large amounts of data. Whether or not like, specifically RNNs trained by backpropagation and all this stuff, that is like the way forward. That I don't necessarily believe that, no. But I think that's where the challenges are going to be, is like what kind of models with what kind of constraints are going to be the right approach. How biophysically realistic should they be? How many parameters can you record in slices and insert into the models and stuff like that? I think those are the questions that we as a field need to figure out. I don't have answers for those. I'm only advocating for the larger general idea that large data sets and explicit models are, I think, the way to go forward.
If you don't mind, just one final question, and this is more of a curveball, I think. Wouldn't you say that even with all the models we have, even if they are more limited than the ones we are trying to develop, wouldn't you say that the primary issue about science, particularly in this field, isn't the fact that we are still very bad at asking the right question?
I don't know if we're bad compared to who. I mean, I guess I think I would say that the hardest part of science is asking the right question. So yeah, so I guess maybe in that sense that we could say that's the thing we're bad at, sure. Yeah, I guess so. I mean, I don't. Yeah, I mean, I think that a big part of what I was getting at in the early part of my talk was trying to make a case about how people were asking the wrong questions because they were limited by their technology, right? And I think that people underestimate the how our technology shapes our conceptual thinking about the problems. And so in that sense, I think I'm agreeing with you. So yeah, I mean, the difference is now we have more techniques and more stuff. So like, the Drosophila, they have a whole brain connectome with Drosophila now, stuff like that. So there's a lot of questions you can ask with that you couldn't ask before. And maybe we can start asking the right questions if we're not too limited by, by just sort of our technology and the journal system and the funding system and the career advancement system and all that. So.
Thank you.
I think that was it for final questions. Thank you so much, Luke, for being here. Thank you for your talk. And then we can discuss on how to send you the class. Yeah?
Okay, cool.
Thank you so much. Bye.